Okay, I hope you all went and watched the syllabus or an introduction to the class video. There you'll learn about how the grading is set up, the discussions that we're going to have, activities, breakdown of the quarter. So please go back and watch that video if you have not watched it yet, and then proceed to this. So we're going to start off with Chapter 2 of the textbook. Chapter 1 is just kind of a brief introduction to genetics. We're going to cover that a little bit here before we actually get into Chapter 2, but really the meat and bones of genetics and what you're going to need to know to pass your MCAT, to go on to whatever biological job that you're looking at in the future. The genetic knowledge we're going to obtain, we're going to start at Chapter 2 for all that. So whenever I've taught genetics in the past, the one thing that always comes up, the first thing that most undergraduate students think of is genetic disease, right? So this is a child that has Down syndrome. This is a chromosomal abnormality, so it's a genetic abnormality. And generally, pre-med students, such as many of you, are kind of conditioned to think of genetics in terms of human genetics and genetics of disease, but that's not all genetics actually encompasses. There's much more to it. Another example of genetics of disease, this really dates me. I asked in my previous lecture when I was at another university if any students knew who this was. I had 130 students and about 30 of them knew who this was. This is Michael J. Fox. Michael J. Fox, if you've seen Back to the Future, Doc Holliday, some Spin City was a show in the 90s. Anyways, he has Parkinson's disease, and so this is another example of a genetic disease. It's not a chromosomal abnormality like Down syndrome. This is actually mutations to genes that cause this, and we'll get to that later. But here's another example of genetics of disease. Another kind of health-related aspect that genetics plays a role in that you're likely to be familiar with is mutations in organisms that affect humans. And so an example of this would be MRSA, which is antibiotic-resistant bacteria. And so you've heard of cases where people don't follow through with the full course of an antibiotic. And what happens is it doesn't clear the disease out all the way, and it selects for bacteria that have genes or mutations in genes that are resistant to that antibiotic. And then at that rate, these bacteria pass down those genes to their offspring, and soon you have a whole population of this bacteria resistant to your antibiotic. And so to get around this, as the doctors tell you, you have to make sure that you take your antibiotics all the way through the course of that they tell you. You don't stop when you feel better, you stop when the course of the antibiotics is run. And doctors also get around this by trying to incorporate multiple types of antibiotics and kind of cycle through antibiotics so that these organisms don't develop this super tolerance to certain like amoxicillin or ampicillin or something else, and it kind of keeps them on their toes, so to speak. So another aspect, we're going to step away a bit from the health aspect of genetics here and look at dog breeds. So dogs, although all these different dogs, we have German Shepherds and Boxers and Boston Terriers and a Beagle, all these different dogs are the same species, right? And so usually when we look at a species like an alligator here in Louisiana or a bear, grizzly bear, a lot of those species look very similar, any individual in the population. But dogs have had this process called selective breeding, where we've taken certain individuals of the population and bred them to other individuals to get a desired trait. And so what we're doing is we're selecting certain genes that are present in these dogs that give them the long, thick coat of a German Shepherd or the stubby nose of a Boxer. And we continue to push that selection to get those desired traits. This is kind of opposed to natural selection, where you have some force in nature that is selecting for the certain trait. So you have artificial selection for dogs, and this also happens in crops. And then you have natural selection, which happens in nature through evolution. Another aspect of genetics that you'll hopefully not have in every day in your life, but you're exposed to now. My wife is crazy about this ID discovery, so are my parents, watching all these crime shows. But DNA analysis for forensics, right? And so now with our knowledge of DNA and selective markers, we can actually link DNA that we find on a cigarette butt or on a drinking glass or some chewing gum or hair left in a vehicle and things of that nature. We can link that back to the individual based on these genetic traits that we know. And so hopefully you don't have this happening to you where you're getting linked back to something, because that usually means you did something bad. But it's also a very useful tool for law enforcement. Another example that you're faced with, probably on a weekly basis, well, every day when you're going to go to the dining hall or to the lunch room, is with food, right? So genetics also plays a big role in the food that you eat. And so not only in aspects of natural selection that we talked about earlier. If you've ever seen wild apples, they look nothing like these Granny Smith apples right there. They're small and full of seeds and not as sweet. And they've been selectively bred over years to be more nutritious and desirable for humans to eat. But also, maybe back in this corner back here, if you can see, there's organic versus GMO decisions that you need to make. And so GMO being genetically modified organisms, these are crops and animals that we've taken, or scientists have taken genes from a different animal or a different organism and put them into a recipient organism. And this is done usually for some sort of beneficial trait, whether it's resistance to a herbicide like Roundup Ready crops like or BT cotton, you may have heard of. One of the first earliest organisms that actually was a GMO and on the market was the Flavor Saver tomato. This was back in the late 80s, early 90s, I believe, where they actually took genes that made the tomato last longer when it was being shipped. And so this meant that a larger amount of tomatoes actually made it to market, and the tomatoes that did make it to market were much firmer, and they weren't getting beat up in the transportation process, and so you had higher yields, and tomatoes became cheaper because of that. So it's not just these medical conditions, but also the food you eat has genetic aspects and considerations to be made. Lastly, we also know that genetics plays a big role in evolution and natural selection. So Charles Darwin, when he sailed on the SS Beagle to the Galapagos Islands and collected finches and looked at all these, the tortoises, the different species of tortoises and iguanas and all these different things on the Galapagos Islands that had these different components, he started to hypothesize that there was some sort of heritable component that was passed down from parents to offspring, and that separation of these subpopulations of finches and things led to development of these different traits. And so these natural processes played a role in selecting for certain genes in certain populations. We're not going to talk a whole bunch about evolution and natural selection just because there are other classes that address that, and other faculty within the department that are much more of an expert in these regards, as I'm a little bit more of a molecular geneticist. But this also is a very important part of natural history and how we got to be here as human beings. Genetics played a big role in these processes of evolution and the development of life on Earth. Okay, so that wraps up chapter 1, really. It's just kind of a broad 1,000-yard view of genetics, and I also thought we could cover that in a few slides just to show how genetics affects you in everyday life outside of the medical component. I know most of you are very excited for being pre-med and to get into medical components, but I also want to give you a whole rounded view of what genetics is. And sometimes genetics creeps up in places that you don't even think about it. For instance, my research at Louisiana Tech, I'm a space biologist, and I use genetics to look at how plants grow in space. so that in the future, maybe 100 years from now, hopefully less than that, we'll be able to grow plants on the moon or Mars or wherever we're pushing space exploration to. And so outside of this medical component, I'm going to try to give you some other examples of how genetics is being studied or affects you in real life. So each chapter, we're going to have this outline at the beginning of the chapter. That's for your reference. So when you go back to study, you can kind of look at these chapter outlines to see where your notes are and where we covered those different topics for the exam. So if I were to ask you to name a geneticist, most students will be able to say something about Darwin, who came up with the theory of evolution. He didn't know about genes at the time, but he knew there were these inheritable forces underlying these patterns of inheritance that he'd seen. Some of you may be able to name Watson and Crick. They are the scientists that came up with the structure of DNA. If you're really into genetics, you might name Rosalind Franklin, who was also part of that team, but didn't really get credit for discovering the structure of DNA. But the real father of genetics is Gregor Mendel. Gregor Mendel was a monk, a German monk, and what he did is he was a hobbyist gardener, we'll say. He studied pea plants. By the end of genetics, this class, you'll probably hate pea plants. You'll never want to see one because they are primarily the example we use for our Punnett squares. And you may have heard the horrors of Punnett squares. But Gregor Mendel was a very observant monk who grew plants and did these crosses and found these simple patterns of inheritance that kind of laid the groundwork for what we know as genetics today. Here's a good look at Gregor Mendel. So Gregor lived back in the early 1800s. He was born in 1822 and he lived to 1884. So this is well before Darwin came up with his theories on evolution. That was the late 1800s, well before we knew the structure of DNA or even what genes were. Obviously, he kind of laid the groundwork for genes. But he was able to make these observations in the plants that he studied to kind of figure out that, yes, there's some sort of inheritable component between parents and offspring that lead to these passing down of traits that we can see. One of the central components to genetics is mutations. So if genes didn't have mutations or didn't accrue mutations over time, all individuals of a species would look exactly the same. All humans would look the same, all dogs would look the same. And while that might not seem like that big of a deal, when you start to factor in the selective pressures from the outside, then that can be a real problem. So we'll take a real silly example here and say that all humans had blonde hair, right? And there's no mutations that changed that. All of a sudden, a disease comes along, or some sort of external stimuli, a toxin in the water or something that is exposed to this population of blonde-haired humans, and it selects and kills everyone with blonde hair. Well, if there's no mutations that have occurred in the population, that means the entire population is going to be wiped out. So these mutations, we always think of mutations as, oh, that gene's been mutated, that's bad, but it also can provide protection from these selective forces. So for instance, if you look at a natural population of humans, we have people with black hair and red hair and brown hair and blonde hair and all sorts of shades in between. And so this wide range of different hair colors or phenotypes, so these traits that we observe are phenotypes, this wide range of phenotypes protects us from our silly example of a disease that kills all people with blonde hair, right? So that population is protected through mutation. And so in this example here on this slide, we have a picture of a Arabidopsis. That is the plant that I use in my lab. And we have 6 different phenotypes of flower that we see here. WT generally stands for wild type. That is the most prevalent phenotype or physical trait that you see in a population. Then we have another one. We have leafy, LFY is the name of that mutation. and a couple of the AP mutations that cause changes in these flower phenotypes that we see. We also see the same trait in bacteria. So it's not just plants and humans, bacteria being a living organism. We see that genetic changes arise in these bacteria too, and this can cause changes in growth behavior of bacteria. So here we have a wild type of this bacteria that is grown, so it grows in what's called a lawn. If those of you that have had microbiology already will know what a lawn is, so it's just completely covered this petri dish. But changes in certain genes cause different growth parameters for this bacteria. This is all the same species of bacteria, but certain genetic changes change how that behavior is achieved in this organism. So to investigate the underlying differences in plant phenotypes, remember phenotypes being a physical trait that you can see, Mendel studied differences in 7 different plant phenotypes or traits. These were pure lines, so remember pure lines, which are traits that were where inbreeding them, or selfing them as we call it, results in offspring which are identical to the parent. So if you take a pea plant that is round, or round peas versus these kind of wrinkled peas, if you take a round pea plant and you self it, or you make it pollinate itself, then all of the offsprings will have that same round phenotype. Similarly with any of these other traits, so purple flowers, if you took a purple flowered pea plant and you pollinated it itself, you would have purple flowered offspring as well. So just kind of, we talked a little bit about self-pollination or selfing, and I just want to make a quick distinction between cross-pollination and self-pollination, because that's going to be very important for talking about Mendel and how we set up these experiments. So in cross-pollination, that's where you take pollen from one parent, and in this example, we use a little paintbrush. Like if we were to do this in the lab, we would take a little paintbrush and get some pollen stuck to it. And then we would take that pollen and transport it into a accepting parent, or a second parent, and pollinate the pistil. But at the same time, we would also make sure to remove the anthers from that plant so it can't pollinate itself. This way we know that only pollen from our donor plant has actually been transported and fertilized the offspring of the accepting parent. Now in selfing is slightly different, where the pollen from the one parent is actually used to pollinate itself. And so some plants, like Arabidopsis, the plant that I work with, will just naturally self-pollinate. You don't actually have to take a paintbrush and collect pollen and put it into the female organs of the plant. But for those true breeding organisms or plants that we saw on the previous slide, those are the result of this, where they've been used, the pollen from the same organism is used to fertilize itself. Let's look at some of the experiments that Mendel did to discover or to unravel these inheritable factors that organisms were passing down from parents to offspring. And so one of the first things he did is he took yellow pea plants and he selfed them. So he took a plant that produces yellow peas and he took the pollen from that to pollinate itself, and he found that the offspring of that self had a predictable ratio of 3-to-1 always, where there was 75% roughly of the offspring had the yellow phenotype that we saw in the adult, and 25 of those offspring, or 25% of those offspring, excuse me, had a green phenotype. So they didn't look anything like the parent. So when we selfed this yellow pea plant, we have a 3-to-1 ratio. Now, if we take a yellow pea plant and a green producing pea plant, and we cross them to one another. So if we take pollen from the green plant to pollinate the yellow plant, or pollen from the yellow plant to pollinate the green plant, we call this a reciprocal cross because you can go either way. We found that, again, there was a predictable ratio for the offspring, but instead of being 3-to-1 like we saw with the selfing of the yellow pea, we get this predictable 1-to-1 ratio, where there's 50% of the offspring are yellow peas, and 50% of the offspring are green peas. And so as you can see in the picture in the pod here, each one of those peas are the seeds, right, for the next generation. So each of those peas represents one individual of this F2 or offspring generation. So in our example, we looked at yellow peas and green peas, and we saw these different ratios that occurred. But what Mendel also noticed is that this was the same apparent ratio for a whole bunch of different traits. And so this 3-to-1 showed up in round and wrinkled seeds, the colors that we talked about before, yellow versus green, the flower colors, purple flowers versus white flowers, the pod color, the length of stems, long and short, and they all showed up in roughly this 3-to-1 ratio. And you have to remember, this 3-to-1 ratio is kind of a theoretical ratio, right? But it doesn't mean that we see that naturally or a solid 100% 3-to-1 ratio. So there's a little bit of other aspects that will affect these ratios. And we'll talk about how some of these changes to fitness and different inheritance pattern affect these ratios. But generally, they're in that 3-to-1 ballpark where there's a little bit of drift away from a true 3-to-1 ratio. For example, 2.96 is real close. But if you have a large enough sample size, you'll get closer and closer to an actual 3-to-1 ratio. So let's look at one of these crosses that Mendel did. We'll take his cross where he took a parental generation that consisted of a pure breeding yellow pea plant and a pure breeding green pea plant and cross them together. Now remember, you can take pollen from yellow and pollinate green, or pollen from green and pollinate yellow. We call that a reciprocal cross. Either will work in this instance. Now, if we look at the F1 generation, the first filial generation, we see that 100% of the offspring are yellow pea plants. Now, if we take that F1 generation and we self them, or we cross them, do a SIB mating, it's called, where we cross one individual from the F1 generation to another individual from the F1 generation, because they all have the same genetics. If we do that, we see that 3/4 of the offspring are yellow, and 1/4 of the offspring are green. Conversely, if we take that F1 generation and we cross it to a green plant, we see that 1/2 of the offspring are yellow and 1/2 of the offspring are green. Now, Mendel had an explanation for this. He said he predicted that each of these traits, for instance, we're talking about color here, has 2 states. And so he denoted these with letters. And so for the yellow pea plant, we give them 2 capital Y's for yellow. And for the green pea plant, we give them 2 lowercase y's. Now, if we cross this yellow pea plant, 2 uppercase y's, versus a green pea plant, 2 lowercase y's, then 100% of the offspring are big Y, little y, or heterozygous. That's because from this first parent, your only option is to get an uppercase Y, because it's homozygous dominant. It only has uppercase Y's. From the other parental individual, you only have a chance to get lowercase Y's, right? This is homozygous recessive. And so that means that 100% of the offspring then are going to be this heterozygous, where they're big Y, little y. Now, if we take those F1 individuals and we self them, we're going to assume equal segregation. What equal segregation means is that when you inherit a gene from your parent, you get a 50-50 shot to get one of those two genes, right? There's not, in this example, each parent is going to be big Y, little y. So you have an equal chance, a 50% chance to get the big Y, and an equal chance, or 50%, to get the small y. So if we line up parent 1 on the top axis, and parent 2 on this, the other axis, we see that we have a 50% shot to get the big Y from parent 1, and a 50% shot to get the little y from parent 1. And you have a 50% shot to get the big Y from parent 2, and a 50% shot to get a little y from parent 2. Now, we do these crosses, and we actually breed them together, and we see that we have 1/4, or 1/2 times 1/2 chance to get big Y, big Y. We have 1/4, or 1/2 times 1/2 chance to get big Y, little y. Again, if we multiply 1/2 chance from parent 1 to get the big Y, and 1/2 chance from parent 2 to get the small y, we get 1/4 chance to be heterozygous again, big Y, little y. And then lastly, you have a 50% chance to get a small y from parent 1, a 50% chance to get a small y from parent 2, and thus a 1/4 chance to get small y as your genotype. And here they've just filled in the 1/4 that I've been drawing here. So for this next example, I'm turning my camera off here so we can actually see what's going on down here. But we're going to look at this second cross that he did, where he crossed the green homozygote recessive to this F1 plant right here that we later denoted as heterozygous. So in this example, we have only a chance to get the heterozygote recessive gene on the top. So they really took a shortcut here, and I kind of don't like it. So if we extend this out, this Punnett square, and say we have small y, and then on the other axis we have big Y, small y, then we have a chance to get, this is a big Y, little y, this is a little y, little y. Sorry, my pictures aren't that great. Here we have a big Y, little y, and here we have little y, little y. So you get a 50% chance, this is 50% to get big Y, little y, and then 50% chance to get little y, little y. So they took the shortcut and said all y at the top. So you get a big Y, little y, and a little y in the way that they did their Punnett square. I prefer you do it the way that I showed you when I drew it out, because it just makes more sense and it's less confusing. But for your textbook, when you go back and look at your textbook and at this lecture and your lecture notes, they just kind of cheated and jumped ahead. Now, one quick footnote to these crosses and all of Mendel's work is that we have to remember that this is only occurring in sexual reproduction. So this only occurs when two organisms are combining their genes to make offspring in the next individual. There are individuals that undergo asexual reproduction. Bacteria is an example. The cells in your body that are undergoing mitosis, that is, they're cells that are replicating in an asexual way and a non-sexual way. And so these crosses don't apply to that, because at that point you're not mixing two alleles from a mother and a father. You're just duplicating what is already in that organism to make an exact clone or replica of that organism or cell. So speaking of sexual versus asexual reproduction, we're going to have a quick refresher here on mitosis versus meiosis. So in mitosis, we have interphase, where there's two copies of the chromosome, one from your mother, one from your father, and this is the same in meiosis as well. During prophase of mitosis, we have replication of those parental chromosomes, and you can see they're lined up here, and each one, your mother's and your father's, is replicated. You have metaphase, where they line up along the pole of the cell. Anaphase, where the spindle fibers come down and attach to the chromosomes and pull apart your chromosomes from your mother and your father. Telophase, where the cell begins to divide around those now segregated chromosomes. And you have two daughter cells that are identical to this first parental cell that we started with. So they've replicated your mother and your father's chromosome and have two of the exact same cells at the end of mitosis. Now in meiosis, we have the same replication like in prophase, where we had our original copy, two chromosomes, and they're duplicated to be 4. During metaphase, instead of lining up along the poles, they line up in pairs. Then, during anaphase, instead of pulling apart the two duplicated chromosomes, they pull apart the pairs of chromosomes. Telophase results in the two pairs being segregated to different cells. Prophase, we have two completely different daughter cells. And then we have a second metaphase 2. So this was actually prophase 2. Now we have metaphase 2, where these segregated chromosomes are now lining along the pole like they do in mitosis. Anaphase 2, the sister chromatids, or the individual copies of those replicated chromosomes, are segregated to the two poles of the cell, where they again divide in telophase. And what happens is we end up with 4 daughter cells that are not full complements of the chromosomes. And so what you end up with is half the genetic information that you started with, right? So at the very beginning we had 2N, 2 copies of chromosome 1, and at the end we have just N, half that much. And think, why would this be important? Well, if you're undergoing sexual reproduction, what you're doing is you're taking half the genetic material from your father, or from a father figure, and half the genetic material from the other spouse, the mother in this case, and you're combining them together to make one full competent genome again. If you didn't use meiosis to segregate the DNA into half, then every time a mother and a father contributes their DNA to a new offspring, you'd be doubling the genome, because you'd have 100% your mom's genome and 100% your dad's genome, so it'd be twice the size in you. And so in order to get around this, or to reduce this doubling of genomes every time, the process of meiosis reduces the genome into half, so you get half of the genetic information from your mother and half the genetic information from your father. So let's look at this process of meiosis again, but instead we're going to focus on the actual genes on the chromosomes as opposed to the chromosomes as a whole. And so in this example here, we have this kind of mustard yellow chromosome and a brown. The color choice is very interesting here for the textbook. So we look at chromosome, the yellow chromosome here, and we have a big A. And so we call this a dominant allele. So an allele is just a version of the same gene. So they could have mutations in different points, One could be dominant versus recessive, and we'll get a little bit more into alleles in a second here. But in this case, we have a dominant copy on the yellow chromosome and a recessive copy on the brown chromosome. And then during meiosis, we have replication of each of these two parental chromosomes. So now we have 4N, right, where we have 2 copies of big A and then 2 copies of little a, each on their own respective chromosome. We have those chromosomes then segregate into different cells, right? And then they line up along the pole and segregate again to the point where we have 4 daughter cells, right? And in each of those daughter cells have one of the 2 possible alleles. Half of the daughter cells have that large A that we started with, and half of the daughter cells have the small a that we started with. So you still have a 50% shot, even though there's 4 daughter cells created through this process, you still have a 50% chance to get your dad's allele or a 50% chance to get your mom's allele. So when we talk about alleles, alleles are just, like I said, they're different. They're the same gene, but they have a slightly different sequence. And so they'll have a mutation here and there. So in this example, we have allele 1 and allele 2. This X indicates a point mutation that has changed the sequence just slightly. So one amino acid might be different. And so that protein might function just a little bit differently on mom's allele than it does for dad's allele. But you can also have more than just 2 types of allele. It's not like your mom and your dad have one of the 2 versions of this gene in the entire world. Within a population, there could be tons and tons of different versions or alleles of this gene, right? So say the gene that makes hemoglobin, right? One of the globin genes. There might be 50-some different versions in a population that are functional, but just have slight little changes here and there, right? And some alleles are more common than others because they provide greater fitness. Obviously, if you have alleles that have a lot of mutations in them from the wild type, then they might not do as good of a job as the allele that is persisted throughout time and proved to be functionally the best allele. But for example, in this figure here, we see that we have wild type allele A, and then we have 3 mutant alleles of it. We denote that with this little tick mark. So we have A prime, A double prime, and A triple prime. And the difference is they're all mutants, but we also want to distinguish where those mutations are. And so A prime and A double prime, while they're both mutants, the mutation's in a different spot. So we need to indicate that those are different because they may have different functionality. They might have different prevalence in the population. So it's important to know that different individuals, even though the gene does the same thing, those mutations cause them to be different alleles, as we call them, and that depending on where this mutation is, makes those alleles different. Now that we've talked about how mutations in different parts of the genes lead to different alleles, we should also recognize that where this mutation is on the gene can have different effects on the gene and thus the protein product of that gene that comes about. And so in this first example here, we have just a wild type gene where we have the promoter. The promoter, if you remember, that is the location where the transcription machinery, so the stuff responsible for making the mRNA, where that binds to start making the mRNA and then later the protein product. We have exons. So exon, that is the region that is actually turned into RNA and then thus turned into a protein. Introns are little regions inside the gene that don't actually get turned into the protein product, so they're spliced out during mRNA splicing. We'll get into all of that stuff later. We have another exon here at the end, and we also have these pink regions that are the protein active sites. So if this protein has some sort of catalytic ability or responsibility where it binds with a catalyst of some sort or another protein, those are the spots in pink are the actual parts of the protein that actually interact with something. And so in this wild type gene allele that we have here at the top, we see there's no mutations. Now, M1 is going to designate our M1 allele. So it's this specific mutation. And we see that there's a mutation denoted by this pink circle in the promoter. And the result is it's null. What that means is that no protein product is functionally active from this gene. So in this case, it's likely that that mutation has caused a lack of ability for the transcription machinery to bind to that promoter to actually turn on or transcribe, make copies of that gene for protein production. In our second example, the M2 allele, we have a mutation in one of these protein or active sites, the binding sites that we had just talked about. And so in this case, we also see that there is a null phenotype, meaning that even though this protein product likely would still be made because there's nothing stopping transcription here, like in the first, the M1 copy, although this protein's made, when it actually gets around to where it has to be in the cell floating around looking for a binding partner or its catalyst to work with, that binding site is screwed up from this mutation, and thus it's not able to perform its function. So we see the same result as we would a change in the promoter where we're actually not getting a functional protein. M3 is just another example. We can, since there's multiple catalytic binding spots on this protein or within this gene, we see that these mutations can happen at any of those binding sites, and we can still get a null phenotype from that gene. So it doesn't matter what binding site the mutation's in, if it's in one of them, it can still have the same effect. Now, whenever there's a mutation in a binding site, that doesn't mean that it always stops the function of that protein. What we can get, such as in this M4 allele, is a mutation in this last binding site, and we get what's called leaky expression or leaky function. And what that means is that we don't have a full termination or full stop of the function of this gene, but instead we have a reduced function of the gene, or leaky function, meaning that it works half the way. It's not as efficient as the wild type, but we see some function, but it's just not as strong as the wild type. And so that's because that mutation there has caused a little bit of a change that leads to this lack of function. In M5, we can have a what's called a silent mutation. And so what this is a mutation that occurs somewhere in the exon, but it's not in the binding site of that gene. And so therefore, there's not a noticeable effect to that gene. It doesn't cause any sort of conformational change of the protein when it's folding or being turned in to the protein. It doesn't have any sort of effect on the catalytic site or the binding sites. And so therefore, it doesn't have any sort of change. There is a mutation there, but it doesn't make the protein any worse off than it was from the wild type. And then lastly, this was an interesting case. It took me a while to figure out what the authors were talking about here because they don't really go over it very well. But generally, if there is a mutation in the intron, those introns are spliced out during RNA processing. And so those introns usually never get turned into a protein. However, in this case, there are little pieces of code, of genetic code, on the boundaries of introns and exons. And that tells the transcription, the spliceosome, this piece of genetic machinery that comes in and cuts out those introns, it tells them where to cut. And so, for the most part, if you have a mutation in an intron, you're not going to see any problem with the protein product from that. However, if your mutation is one of those very small 2 base pairs that designate the border of an intron and an exon, then that can cause the spliceosome not to recognize that spot. And so therefore, this one specific case, if there's a mutation on the very edge of an intron, you can end up with a mutation that causes a null mutation. And what ends up happening is that that border that I talked about, where we're saying this is the border of the intron and the exon, that information is lost. And so this entire piece of intron, which is supposed to be cut out, is then incorporated into the gene, and then the gene is completely different shape than it's supposed to be. If this seems a little bit deep right now, don't worry about it. We're going to get into this in a later chapter. But really the takeaway from this is that mutations in different parts of the gene can lead to different results. We call those different alleles. And mutations at different places can also have the same result as another place. Depending on where you mutate, you can have a bunch of different locations that lead to null mutations. So when we talked about our inheritance of chromosomes, we failed to mention the role that sex chromosomes play into it. And so not every organism has sex chromosomes, right? So bacteria and things of that nature. We'll get into plastids a little bit, but they generally don't have sex chromosomes. But in humans, obviously, males are XY, and females have 2 copies of the X chromosome, so they're XX. And the majority of these genes on the X and Y chromosome are different. At least in the case of the Y chromosome, a lot of that are sex-specific genes for creation of males. If you want to get really into it, look to take my developmental biology class that I teach, and we'll get real into the weeds of sex determination and development. But this also affects our Mendelian inheritance patterns. And so depending on if a gene is on an X chromosome or Y chromosome, since those genes are different on the 2 chromosomes, we see it affect our inheritance pattern. So one of the ways, a simple way to look at inheritance of, or sex-linked inheritance, I should say, is through the study of Drosophila melanogaster. If you're a college student, you probably have had these in your kitchen at some time. They love old beer. I had an infestation when I was an undergraduate, but these are fruit flies in essence. And so you see them around. This is a close-up. They actually look like a fly when you get close, but when you see them in your apartment, you know, there's a little speck flying around. But they're played a monumental role in determining a lot of genetic traits between crossing over effects, which we'll talk about later, and also sex-linked inheritance. And so what makes them a wonderful model is that they have a short life cycle. And so you go from adults to adults in about 5 days. So you can get multiple generations to study this inheritance by breeding them. They're really easy to take care of. They eat a lot of, you can feed them potato paste and stuff in the lab, like dehydrated mashed potatoes, and they love that. And so they're a great model for determining inheritance. So what we're going to look at as an example of sex-linked inheritance is the eye color. So we have 2 phenotypes. We have white eyes and red eyes in Drosophila. And so we're going to look at 2 crosses associated with this eye color. So in our first cross, we see that the eye color gene, we have 2 alleles. We have the red allele, which is W+, and then the white allele, which is just W. And we see that that's only on the X chromosome, so it's not on the Y chromosome. So if we take a homozygote female, so it's got 2 copies of the red female, and we cross it to a white male, and conversely, we do a second cross where we have a homozygote white female and cross it to a red male, we have different outcomes. So the product of that first cross, our F1 over here, we see that from the male, we can get the white copy only on the X chromosome, because remember, there's no copy on this Y chromosome. We see from the females, we get 2 copies. We only can get that red copy from the females. And so the product of this cross is 100% red offspring. So we have half of them that are female and half of them are male, but they only could inherit that red gene because that white gene is recessive, and it can only be inherited in this specific in females, right? Because the females are the only ones that get this X chromosome from the father. And so those females, because red is dominant, they have a recessive copy of that white. So 100% are red offspring. In our second cross, where we have the white female and the red male, we see that on the male side, you can only donate this red copy. And the females are donating 2 copies of the recessive white, right? So in the females that have 2 copies of this X chromosome, we see that they are always red because females can only get that red copy from their father. If they got the Y, then they become a male, right? So all the males, though, are white because in order to get this Y chromosome, you had to also have only gotten a white chromosome from your mother, so you have XY to become white males. Now let's look at selfing of the F1 generation. So if we take, in this first example, the red female and cross it to a red male, we have the male up on the top here and then the female down at the bottom here. We see that we get that familiar 3-to-1 offspring ratio, right? And that was similar to the selfing that we saw Mendel do with those pea plants. Now, if we take the second cross, where we have white males, and we cross them to these red heterozygote females that we put over on this axis, we see that we get that similar 1-to-1 ratio that we saw from the pea plant that was green, the test cross, the homozygote recessive that we saw in Mendel's example. So sex-linked inheritance, the genes, if they are on sex chromosomes, can also change the expected ratio of inheritance depending on if the male is red eyes or the female is red eyes, or vice versa, or a mixture of white and red. Lastly, so I'm going to put onto Canvas lists of vocab. And these are good terms for you to know, specifically because they're going to help you read not only the textbook and understand the lectures, but also when you look at problems and things of that nature. I want you to be able to read a problem that says we take a pea plant that is homozygous green and self it. Well, you need to know what selfing is to be able to answer the question. So generally at the end of each lecture, and specifically I'll put online these vocab words that I want you to know specifically from that chapter. So thanks a lot for taking the time to listen, and check out chapter 3.